Then let me start. Then thank you for all the people that are watching this presentation. It's for me an honor to present you Martin Wake. Martin Wake, I learned something very interesting because he born in Switzerland, but he's a German citizen. He did his PhD at Grenoble, the Institute de Biología Estructural, in biophysics. After that, he did 2 postdocs, one at the Big Boy Center in Utrecht in the Netherlands, and the second one at the Weizmann Institute in Israel. Currently, he is the chairman of the research group Dynamics and Kinetics of Molecular Processes at the Institut de Biologie Estructural IBS in Grenoble, France, as well as visiting professor at the University of Parma. Martin is author of more than 100 papers and have a lot of citation of his works. Then thank you very much for accepting the invitation, Martin, and please show us the difference to have different strong sources of different energies to describe proteins.
Yeah, let me just check that this is working. Can you see the right configuration of my presentation?
Yes. Yes.
Okay, good. Then I first would like to thank you and Enrique, and thank you Nina, and thank you Carlos for having invited me to Mexico, at least virtually. It's a pleasure for me to share today some considerations about time-resolved X-ray crystallography at both synchrotrons and X-ray free electron lasers. So I first would like to show you where I'm sitting at the moment. I'm sitting at the moment in France, in Grenoble. Grenoble is a nice town in the middle of the Alps, and we have quite a huge research campus, the European Photon Neutron Campus in Grenoble, that hosts 2 large-scale facilities: the synchrotron of the European Synchrotron Radiation Facility, the ISRAF here, you see the ring here, and the neutron reactor of the Institut Laue-Langevin. And I'm sitting here in the Institut de Biologie Structurale, and I just found out together with Carlos that he was working here and me also quite a few years back in the same institute. So the proximity of our Institute of Structural Biology to these large-scale facilities explains why our research on the molecular biophysics mostly uses these X-ray and neutron sources. But let me first show you when and why I met our chairman for the first time. That's actually almost 20 years ago. Of course, we didn't get older at all, as this photo testifies. And we have done, together with Elspeth Garman from the University of Oxford and Robin Owen, who is now one of the beamline scientists at the Diamond Light Source in the UK, we have done many experiments, and it was at the ISRAF trying to work out what radiation damage does to your crystalline proteins and how can we possibly mitigate it. And together with Enrique, we had spent many days and nights at the synchrotron, having also a lot of fun celebrating birthdays, as you can see on this photo. Our research is concerned with studying the structure and the function of proteins, as I think all of you do, if I understood Enrique correctly. Now, we as many others, we include a dynamical dimension in our consideration, because we all know and realize, of course, that in order to understand protein function, we need to understand this delicate balance in this vital triangle between structural, functional, and dynamical aspects. And of course, we know protein dynamics happens on a very large time scales, all the way from femtoseconds, where we have electronic transitions, enzymatic transition states, bond vibrations, such as, for instance, here, the vibrational modes here in this water molecule, or on a picosecond, nanosecond time scale, where we see side chains move, such as those involved in water gating through aquaporin membrane protein. On a slower time scale, microseconds, we have domains that move. So for instance, here, this MD simulation in which a drug molecule samples different binding grooves on the surface of a kinase. And then on the longer time scale, where milliseconds, seconds, where proteins are synthesized and folded, such as shown in this long MD simulation by the Sol group shown here. So protein dynamics happens on a very broad time scale, and a large arsenal of biophysical techniques have been developed that each open a very specific window to study protein dynamics on a particular time scale. And today, I would like to focus on what X-rays can contribute to study protein dynamics. In particular, time-resolved crystallography, or maybe more broadly, kinetic crystallography, carried out at X-ray free electron lasers on the fast time scale, and on a bit shorter, longer time scale, carried out at synchrotrons. Now, X-ray free electron lasers, as I'm sure you all know, are large-scale X-ray facilities similar to synchrotrons in size, at least. So shown here is the X-ray free electron laser, or XFEL Sakla in Japan, next to the Spring 8 synchrotron. And with time-resolved crystallography or kinetic crystallography at these large-scale facilities can recover protein motions from sub-picoseconds here all the way to seconds here. So in the next half hour, I would like to share with you a few considerations on this time-resolved X-ray crystallographic methods and what we can do with it. But first, have a look at the conventional crystallography. I'm sure most of you are familiar with that. We need an X-ray source, and still in structural X-ray crystallography today, in most cases, the X-ray source is a synchrotron. You shine synchrotron light on a relatively large crystal that you cryo-cool to mitigate at least some of the X-ray radiation damage. You turn the crystal, you collect many diffraction images, in most cases on a single crystal. Together with the intensity and phase information, you you calculate electron density, in which you model and then refine a protein or macromolecular model, and then you get these nice colorful models that we all are familiar with. Now, we all know, of course, that crystallography is an ensemble method. So we have here a crystal with identical molecules, well, almost identical. Some flexible regions, such as the legs here, the loops, they're in different conformations. So then in the end, the structure that we determined, of course, is a time and ensemble average picture of a seemingly static protein. So some rigid parts are well defined, like this guy on the horse here, but then the legs here, the loops, they're sample different conformations. So kinetic crystallography is aimed at obtaining structural snapshots of proteins at work. For instance, structural snapshots of enzymes at work. And let me first remind us that kinetic crystallography works because actually most proteins are biologically active in the crystalline state. And this is because the protein crystals contain a lot of solvent, on average 50% in a crystal. And the protein crystals make only a few crystal contacts. The result is then that ligands, for instance, can diffuse freely through solvent channels. The proteins, they remain flexible. And in most cases, actually, as I said, active, biologically active. But in the crystal, often the reaction rates are slowed down, either because we have precipitant and other buffers, or because of the packing of the crystalline molecules, we get restricted motions in the crystal. But in most cases, crystalline proteins are biologically active. That's an important point to make. Now, kinetic crystallography can be broadly divided into two big groups, either cryotrapping methods where you try to trap intermediates, or time-resolved crystallography, in which you follow non-equilibrium protein motions in response to an external perturbation. So in cryotrapping of intermediates, you collect data at temperature, crystallographic data at temperature well below room temperature. You use monochromatic X-rays, and you can use a home source or a synchrotron source. In time-resolved crystallography, as I said, you follow non-equilibrium protein motions in response to external perturbations. So really, you try to follow a process in real time. And I will focus in the next 25 minutes on this approach in kinetic crystallography. So time-resolved crystallography requires synchronization. We reminded us of the fact that crystallography is an ensemble method. So if you want to follow structural changes in the crystal in a protein crystal over time, we need to synchronize the trigger so that all molecules in the crystal start their activity at the same time. So in time-resolved crystallography, we need to trigger activity, and what methods you use to trigger activity depends on your system and what you want to study. In most cases, you use UV visible light illumination for trigger because it's most convenient to use. This works on, of course, on inherently light-sensitive proteins, or in proteins that have been rendered light-sensitive through use of so-called cage compounds. You can also trigger activity by diffusing small ligands, molecules, for instance, diffusing substrate into a crystalline enzyme and follow substrate turnover. Or you can change the pH suddenly, or change the temperature. For instance, through irradiation with an infrared laser, you get a temperature increase of 20, 30 kelvins, and you speed up a reaction. Or you can also use X-ray radiation itself to trigger activity, or terahertz irradiation electric field. So you see there are many ways to trigger activity in a crystal depending on the protein or biological macromolecule in general that you study. So once you triggered activity in your crystal here that is in its starting resting state, the blue state, you wait for a well-defined but variable time delay, the pump probe delay, and you acquire an X-ray image. And you do this trigger probe process many times with a cell with the same delta T until you have collected enough diffraction data so that you can determine the intermediate state structure that has been populated after delta T1, so the light blue state here, and that you can determine this intermediate state. Then you change the time delay, delta T2, and you repeat the experiment. And then you get this intermediate state, and so on and so forth. So in the end, we ideally would like to have a kind of molecular movie. But in reality, since often just getting one snapshot is a whole experiment in its own, it's rather just a few snapshots put together rather than a movie. But in the future, we very much hope that to get closer and closer to really watching proteins at work with time-resolved crystallography. So time-resolved crystallography, you can do it at the synchrotron or at X-ray free electron lasers. So just without going into any detail, I just want to mention a few things about X-ray free electron lasers. The biggest difference between an X-FEL and a synchrotron is that the undulators that cause or force the electrons that generate the X-rays forced to undulate. These undulators that generate very highly intense X-ray radiation. These undulators in the synchrotron are quite short, say 2 meters, but in the X-FEL they're more than 100 meters long. And because electrons then undulate through this 100-meter long undulator, it generates through a process that I don't want to discuss, just to throw out the name, through self-amplified spontaneous emission, SASI, self-amplified spontaneous emission, you generate extremely bright and extremely coherent X-ray light at the end of this long undulator here. So that in the end, you're left with extremely short pulses in the X-FAL between, say, 10 and 50 or 60 or 70 femtoseconds. And most importantly, you end up with pulses whose peak brilliance or peak intensity is 10 orders of magnitude above the one of synchrotron pulses. And you generate light of very high coherence. That's why it's called a laser, a free electron laser, X-ray free electron laser. So the most important property for us when we talk about time-resolved crystallography is the pulse length. That is 10 femtoseconds, as I said, roughly at an X-FAL, and that is only 100 picosecond in a synchrotron. It's limited to 100 picosecond in a synchrotron because the electrons that are revolving in the synchrotron storage ring, they also come in packages, and these packages are 100 picosecond long. So this actually, the X-ray pulse length, together with the pump pulse length, if we trigger activity with light, if we work on light-sensitive proteins, so the length of both the pulse and the probe, of the pump and the probe pulse lengths, they determine the time resolution of our crystallographic experiments. As I said, 100 picoseconds for synchrotrons, 10 femtoseconds for X-ray free electron lasers. Now, one important thing to consider when you do time-resolved experiments, in particular in light-sensitive proteins, is that light needs to penetrate the crystal. And protein crystal is often optically very, very dense because in a protein crystal are packed, proteins are packed very, very, very densely. So if you want to trigger synchronously in many, many proteins in the crystal activity, as I showed with the horses earlier, the crystals shouldn't be too thick. And I just took here an example of a fluorescent protein crystal that we work on. And you see here, so we look at a transmission of light through as a function of the position in the crystal. And we see that after, in this particular case here, after 3 micrometers of traversing through a crystal, only 30% of the photons are still available. So imagine, take a 20 micron crystal of this fluorescent protein, you activate only the surface of the crystal, but not the interior. So depending on the protein concentration and the photophysical parameters of your chromophore, you may need to work with very, very tiny crystals. And this is one of, I look at this 3 micrometer crystal, that might be much too small a sample to give you useful diffraction information at many or most of the synchrotrons. So when we consider using X-FALs or synchrotrons for time-resolved crystallography, do we need to consider the time scale that we target? So fast time scale, we go to X-FALs, slower time scales, we go to synchrotrons, but we also need to make sure that once we've chosen the right sample size, that we do get a large enough diffraction signal to record. Okay, so at synchrotrons, I think many of you might have heard of so-called Laue crystallography that has been used over the past 30 years to study proteins at work. It uses polychromatic X-rays and is, as I said, reaches a time resolution of 100 picoseconds. In Laue crystallography, you expose a static crystal to wide bandpass X-ray beam. And for those who remember the Ewald sphere construction for Laue geometry, this means that we have all those Bragg spots here, all those spots in reciprocal space in diffraction condition between this sphere corresponding to the large end of the X-ray wavelengths bandpass, and this to the minimum here. So with a single, in a single shot, in a single diffraction pattern, we get many more Bragg peaks than with monochromatic X-rays. So we need actually fewer images to collect the full data set. And that's how a Laue diffraction pattern looks. And we can record a full diffraction pattern in only 100 picoseconds at the synchrotron, because we have so many X-rays in this white or pink synchrotron pulse. So here we have 10 to the 10 photons per 100 picosecond pulse. But you see the Bragg spots, they're extremely closely together. And actually for most proteins, it is difficult, if not impossible, to work with data where the Bragg spots are so closely packed together. So I show you a successful example in which Laue crystallography at a synchrotron has been used to get time response information on myoglobin, the model protein to study conformational changes. And here the researchers, they have collected Laue data at 2 pump probe delays, 3 nanoseconds and 360 nanoseconds. Shown here are so-called difference Fourier maps, where you subtract the diffraction data of a data set collected with trigger light from a data set without trigger light, and you visualize the differences. So in red is a negative difference electron density, so where electrons should disappear after a certain pump probe delay, and in green, where electron density comes up after a certain pump probe delay. So this pair of negative and positive peak, for instance, around the heme group within the myoglobin, means that after this pump probe delay of 3 nanoseconds, the heme group shifted upwards here. And here they could see that the CO that was bound in the dark state structure here close to the heme iron has moved here after 3 nanoseconds and has diffused around the whole heme in 360 nanoseconds. Okay, so this was a successful example, but I think it is fair to say that time-resolved pump probe Laue crystallography at synchrotrons is really limited to only a few very, very nicely behaving, well-diffracting model proteins. And this is because you can first only study cyclic reaction, because you have to pump, bring the protein back to the starting point, pump, collect data, back to the starting point, and so on. And this bears also the big risk, the problem of radiation damage, because radicals that you generate by collecting your diffraction data, they spread on a time scale that is actually close to the time resolution, 100 picoseconds. So by collecting more and more data, you damage your crystal more and more. And the most important reason why Laue crystallography can't really be broadly applied to study protein dynamics in real time is because you require crystals are extremely radiation resistant, and they need to be extremely high quality, so that the mosaicity of the crystals that determines then how closely or how much the reflections overlap needs to be very, very small. Also, the proteins need to pack into very, very small unit cells, again, so to minimize the overlap of the reflections. So all these reasons together made Laue crystallography feasible, but not really successful. Okay, at X-ray for electron lasers now, because we have so many photons in a single pulse, and we use quasi-monochromatic X-rays, well, at least less polychromatic than in Laue crystallography at the synchrotron, and because we can use very small crystals, we can now reach a time resolution of 10 femtoseconds. So as you may know, today there are 5 full-scale X-FELs operational in the world. There's the very first one that started operation in 2010, the LCLS in Stanford in California. Then the second was the SACLA X-FEL in Japan, followed by PAL X-FEL in South Korea, the Swiss FEL in Switzerland, and now the Europeans also have an own machine, the European X-FEL in Hamburg. And there's also in a pilot project at the Arizona State University, a very compact X-felt source that is only a few meters long under construction. And we'll see if that works. But so far, X-felts are really large-scale facilities that are several kilometers long. Okay, so we'll mostly use so because the X-felt pulses are 10 orders of magnitude more intense than a synchrotron radiation, can we work with very, very tiny crystals? We can work with very, very tiny crystals, but because we put so many X-rays into our sample, the sample literally explodes extremely rapidly. And this is a simulation that has been carried out by Richard Neutze and Janusz Hajcu and their colleagues more than 20 years ago, and they termed this approach to data collection diffraction before destruction imaging, because you shine very bright X-felt pulses on your sample and you image it before the sample had time to respond and therefore explode. Because remember, radicals spread on a picosecond, nanosecond timescale and therefore lead to specific damage, to disintegration of the sample. But since the X-felt pulses are so short, say a few tenths of femtoseconds, you can actually collect data before the sample responded. And that's the diffraction before destruction imaging principle, according to which data are collected at an X-ray fractal laser. So in the beginning, when these simulations had been done here, the biggest promise and the biggest carrot for building those X-felts was actually the dream to be able to image individual protein molecules, both in space and time, coherent diffraction imaging. And now after more than 10 years of operation, it's clear that this dream has not come true yet, because there's still not enough photons in a single X-felt pulse. So this is one of the latest papers here, where of this quite huge virus here, they could determine the structure from a single virus, or a single stream of virus at 70 angstrom resolution. So you see, we're very far from reaching atomic resolution on even larger protein complexes. So that's very limited here, and it's really still not clear if in the future, even with increased fluxes at X-felts, if we can get to atomic resolution if we use only single particles. But what had been proven to be very successful over the past 10 years is so-called serial femtosecond crystallography, and I will show you briefly the principle of that approach. So remember, in the beginning, I showed you conventional oscillation crystallography. A crystal at synchrotrons, you use a big crystals, you cool it to cryo temperature, you rotate the crystal, and you collect a series of images from which you know how they're related in reciprocal and direct space. Now, in serial crystallography, at least at X-ray for electron lasers, because each microcrystal is immediately after data collection turned into a plasma, you need to replenish constantly the sample. Because one microcrystal, you get one diffraction pattern. And you need actually many thousands, tens of thousands, sometimes millions of diffraction patterns to get a full data set. So you need to, because of the requirement to replenish the sample constantly, this is called serial crystallography. Whereas most crystallographic data are at a synchrotron are collected at prior temperatures, the serial crystallography approach at X-fold is inherently a room temperature approach. So it preserves the conformational heterogeneity of a protein. That is, of course, restricted if you collect data at 100 Kelvin. The serial crystallography requires new sample presentation methods, different detectors, and data processing and analysis protocols. And this has all been developed over the past 10 years. So sample presentation is very different. You broadly group it into two different families. Either you inject microcrystals, either with a very fast liquid jet or with a slowed down, what people call two-phase injector that carries the microcrystals to the X-felt beam, or you spread many microcrystals on the surface of a solid support, and then you raster scan across the surface of the sample holder. And there are also fancy hybrid methods where you deposit microcrystals in drops on a conveyor belt that then transfers your microcrystals to the interaction region with the X-ray beam. Okay, so not so many years ago, actually, there were 5 applications of serial femtosecond crystallography that were believed not to be easily done, or actually not at all be doable at a synchrotron. And these were the following 5. Study of microcrystals of fragile proteins. I told you, small samples can be conveniently studied with these highly intense X-ray crystals. X-ray beams, sorry. In vivo crystallography, so nanocrystals that grew in cells and that can be actually used for crystallography within the cells. Room temperature crystallography that, as I said, preserve conformational heterogeneity. Completely X-ray radiation damage-free structures, because you collect data before damage actually develops, and time-resolved experiments, in particular on the fast time scale. So clearly, development of serial femtosecond crystallography and X-ray free electron lasers, they have greatly benefited from decades and decades of experience and developments at synchrotrons. But now, over the past 6 or 7 years, I would say, where we observed the reverse development, that everything that has been developed, or many things that have been developed at the X-FEL, now benefit synchrotron crystallography. And I started listing here a few years ago all those papers that use serial crystallography at a synchrotron to collect data, and I stopped in 2019. So serial synchrotron crystallography is so useful or so successful because it distributes the absorbed X-ray dose over thousands and thousands of microcrystals, and therefore minimizes the X-ray radiation damage. So let's remind us, we in classical rotation crystallography, we rotate a crystal, we collect while rotating diffraction images, and the X-ray dose increases while we collect data. In serial synchrotron crystallography, we collect only a single image on one spot. Then we move to a different spot that has not been exposed before. We collect another image, and we do that many 10,000 of times. And therefore, we can compose a very low dose data set in which we minimize radiation damage. And that's the heart of the success of serial synchrotron crystallography, because it minimizes radiation damage. And this work we have been actually done with a colleague that is both dear, I guess, to Enrique and to myself. And I really have to apologize, Enrique, that we managed, much to our pleasure, to stabilize Eugenio in France. And he got extremely good training and experience with you. And we are extremely happy to be able to continue working with him here in France. So he has done great work, and this is one of the examples of his achievements. So because of this development of serial synchrotron crystallography, today it's more and more of the experiments that 5 years ago had to be done at the X-ray fractal lasers can now be conveniently done at the synchrotron. Small crystals, microcrystals even, can now be used at the synchrotron because the synchrotrons get upgraded, they get more and more intense. You can do in vivo crystallography at the synchrotron and also room temperature crystallography. So probably the niche, the small niche that will remain over the past, over the following years, really reserved to X-FELs, is if you need to determine a completely damage-free structure of a radiation-sensitive protein, and I will show you an example, or if you want to do time results experiments, in particular, but not only on the very fast time scale. Okay, this is what I just mentioned now with the 4th generation synchrotrons, and the ISREF synchrotron in Grenoble in France is the first example of which you get 100 or 1,000 times higher flux. That means with monochromatic X-rays, you can do serial or multi-crystallography and hope to get over the following years to a time resolution down to the microsecond and millisecond using monochromatic X-rays. So that what will most probably happen over the following years is that if you want to do time-resolved crystallography, if you are interested in motions on the microsecond, millisecond timescale, you can go to synchrotron. And if you're interested in faster timescale, you go to NIXFOL. So both sources will probably be very complementary. And these are the first papers actually of researchers who have used time-resolved serial crystallography at a synchrotron very successfully to study protein dynamics, here in this case light-sensitive proteins. I told you earlier that synchrotron Laue crystallography over the past 30 years was successful only in a handful of model proteins. But since time-resolved crystallography at IXFOL started in less than 10 years ago, we see here these are all papers that have been published over the past few years, where researchers use time-resolved crystallography to study proteins other than model proteins. And we find here phytochromes, reaction centers, rhodopsins, photolye, an example from last month. So it looks like this method is rather useful to study proteins at work. And as I said, because of the IXFOL pulses that are so short, these new sources allow you to get into the photochemical timescale. And then I would like to illustrate what you can do with high-speed movies of proteins here, exemplified with what happens if a tennis racket hits a ball. I was very astonished when I saw on YouTube what happens when you follow this with a high temporal resolution, that this tennis ball gets so squeezed. I didn't know at all. So by being able now to look at a photochemical timescale at proteins in real time with crystallographic methods, we are really excited that we can see things that we couldn't see before and that we couldn't have guessed exist before. So in our group in Grenoble, we spent the last 6 or 7 years to study photoisomerization in fluorescent proteins. And I don't want to show you details on this, but just summarize it on one summary slide. What we have done here is to study isomerization of this chromophore within a green fluorescent protein type fluorescent protein that can be switched upon light illumination from one chromophore isomer cis to another trans, back and forth, going on and off in terms of fluorescence. And this photo switching happened so fast that until recently, only simulations were available that showed which trajectory the chromophore takes. And what we have been able to do is to capture isomerization intermediate 1 picosecond after light illumination. Here you see the chromophore is twisted, completely twisted in an electronically excited state between the starting trans state and the ending cis state here. So that's a type of structural information that you can now get on this ultra-fast timescale. But what I would like to finish my presentation with is another recent example on a light-sensitive protein, and in particular a photoenzyme. And because there are not so many photoenzymes that have been discovered so far, there are only 3. I hope that you appreciate these few slides here. So we, with many other colleagues together over the past 3 years, have worked towards understanding the molecular working of a newly discovered photoenzyme that actually has been isolated from hydroalgae and that transforms lipids to hydrocarbons in a single step upon illumination. So it turns lipids into hydrocarbons when it's illuminated. It's a photoenzyme, so that means this enzyme requires a photoenzyme for each catalytic turnover. The catalytic activity can be conveniently triggered by light, so that you can easily do studies on very short timescale. And I mentioned already that this is only a 3rd photoenzyme discovered so far in nature. The others, 2 being photolysis, DNA photolysis that repair certain types of DNA damage, and peptide oxidoreductase a photoenzyme involved in chlorophyll synthesis. So here our aim was to understand photocatalysis at a molecular level, and I will show you in the next few slides what time-resolved crystallography could contribute to better understand this enzyme. This enzyme, by the way, has potential applications because it can so easily transform lipids into hydrocarbons. So the idea here of those scientists who are interested in applications is to have large ponds of microalgae and transform in a single step by expressing a lot of this protein photoenzyme, transforming lipids into hydrocarbons that are then very close to what you would use in fuel. Okay, so this is the consortium that worked on trying to understand this inner working on this photoenzyme. We have different groups in this consortium led by Frédéric Besson from the south of France. The starting point of this consortium was a low resolution 3.2 angstrom crystal structure of this photoenzyme that showed that the FAD cofactor, that is the light harvesting moiety in the photoenzyme, is in the heart at the end of a hydrophobic tunnel, in which we can see a substrate coming close to it. A fatty acid, you see this green worm-like thingy there. Now, the first thing our colleagues have been done, modify engineer the protein so that the crystals diffract to higher resolution, and they could actually determine a high resolution, better than 1.8 angstrom crystal structure, using synchrotron cryocrystallography. And the interesting observation here was that this FAD molecule was not planar but bent. And since from biochemical studies it was clear that this starting FAD redox state is oxidized, everybody expected that oxidized, which is normally planar and not bent, would also be planar in this enzyme here. Since it has been, I can illustrate it here. So how in this photoenzyme here, FAD is getting oxidized, FAD is getting activated by light, goes into an electronically excited state. It then transfers an electron, it then receives an electron, the FAD, from the substrate that gets decarboxylated in a so-called forward electron transfer process, and then it gets semi-reduced. And later in the process, once this carboxyl group has been the substrate has been decarboxylated and the radical has been generated here, is the electron back transferred from the FAD back to the alkyl radical, and then it gets oxidized again. Now, as I said, oxidized FAD should be planar. Only the semi-reduced is bent, so that we observed in the starting state a planar oxidized FAD was very strange, and we suspected, as it had been described in other FAD containing molecules in the literature, that this bending is actually due to X-ray induced reduction. So you determine your structure, you generate lots of photoelectrons, secondary electrons, they reduce the FAD and then bend it. That was our suspicion. But then Antoine Royan at the ISREF, he tried very hard and minimized the X-ray dose and could collect a data set at a quite low dose. This probably doesn't tell anything to you, except for Enrique and other aficionados of radiation damage studies. That's quite a low dose, and still at this low dose, the FAD was bent. So we thought, maybe it's not X-ray irradiation. Then Antoine Royan, he operates a spectroscopy lab at the ISREF that allows you to, at the same time, collect crystallographic data and spectroscopic data. So what he did here, he followed X-ray induced photoreduction and determined the half dose to be only 40 kilogray. So you compare this dose at which half of the FAD molecules are actually reduced by X-rays to the dose it takes to collect the full data set. And you see, even if you're very, very careful, you end up with half of the FADs being reduced. So this is one of the typical examples where it's extremely difficult at a synchrotron to get a completely radiation damage-free structure. And here, I mean, we're looking at the heart of the protein, the cofactor, the chromophore. So if you're not sure if the conformation of the chromophore is affected by radiation damage or not, this is a very good case to wonder how we can really get a completely radiation damage-free crystal structure. And that's why for definitive answer, we needed to collect a radiation damage-free 0 femtosecond crystallography structure at the Nixfall, where we collect the data much more rapidly, as I said, before damage had the time to develop. The second aim, when we used X-cells to study this photoenzyme, was the question: when does this decarboxylation of the fatty acid occur? I don't want to go into any detail here, but based on the first studies, there were two scenarios possible. Either the CO2 comes off at the same time that forward electron transfer from the substrate to the FAD, scenario 1, or in the second scenario, it comes off only when the electron is back transferred from the FAD to the alkyl radical. So very simply, we thought, well, if we could determine intermediate state structures after 900 picoseconds, because this forward electron transfer takes 300 picoseconds, and after 300 nanoseconds, that's the time it takes 100 nanoseconds for electron to be back transferred. Well, we'll well see, is there decarboxylation happening here or here? So we could distinguish between these two scenarios. That was our second aim. So you see in this example here, and that's one of the reasons why I picked it, by using seal crystallography at the Nixfall, we can use both, or we can exploit both characteristics of X-file radiation and do studies that cannot be done at a synchrotron. Determine damage-free structure to see if the FAD is bent, and do a time-resolved experiment to see when the substrate is decarboxylated. So we went almost 4 years ago to the LCLS in Stanford with quite a big group here. I should mention that the experiments that our group in Grenoble does are only possible because we collaborated with Ilme Schlichting's group at the Max Planck Institute in Heidelberg. She's one of the few pioneers worldwide who developed hard and software and experimental approaches to do crystallography at X-file. So with her group, we have been able to do experiments. As I said, X-file is a large-scale device, 5 kilometers long. You end up sitting here under the earth in a tunnel and do experiments. And it's quite a lot of pressure because the whole machine, the whole facility only works for you. So you better not forget your sample, or you better are well prepared. X-file experiments are actually quite challenging because they, well, depending on where you come from, they involves lots of traveling and not so much sleep when you work day and night for 5 days often. Depending where you go, there are quite some communication challenges, I must say, and you eat funny food for about a week. So just in one slide, how is the experimental setup? You need many crystals, as I said, microcrystals. You inject them into the X-file beam, and that sounds very easy, but it's actually quite difficult. Those two gentlemen in the Ilme Schlichting groups, they are world expert and know how to do this. You collect, you excite your photosensitive protein with a pump laser, and then after well-chosen pump probe time delays, you collect X-file, you collect diffraction patterns. I will skip, in the interest of time, this slide here and come to the results here. So we have been able to determine the radiation damage-free dark state structure of these photoenzymes. And this was actually solved from diffraction data that have been collected without any pump laser illumination, of course. So what you see here is a zoom into the heart of this photoenzyme, where we force this FAD chromophore to be planar. And the crystallographers among you will recognize in this FOFC map that the map tells you that's not the right conformation, it should be bent. And indeed, if we model the electron density with a bent model, it fits very nicely. So that's really a definitive proof in a completely radiation damage-free structure that very strangely, this oxidized FAD in this photoenzyme is bent. Now, our colleague from Moscow State University, she's an expert in quantum chemical calculations. She wondered, so why is this FAD bent in this photoenzyme? Does it has any role, any functional role? And she came up with two effects that this bending has. The first is that the bending reduces the energy of the FAD radical that is generated once an electron is transferred from the substrate to the FAD, and this facilitates forward electron transfer. So that's one functional role of this bending. And the second is that by bending the FAD, the absorption maximum shifts. It shifts away in this microalgae from the bandwidths where chlorophylls absorb and eat up all the light into a green gap where chlorophylls do not absorb. So this bending, most probably, is a way the enzyme found to capture most efficiently light by getting away from the chlorophyll absorption. And then I told you our second aim want to use time results information to see at which time step decarboxylation of the substrate occurs. And I told you that we have been mostly focusing in 2 time points, 900 picosecond and 300 nanoseconds. And you see again in these so-called Fourier difference maps that decarboxylation evident because of the red negative electron density is already evident at 900 picoseconds. So that means that decarboxylation happens during this step and not only later in this step. So we could actually tell when this decarboxylation in the photocycle occurs. And this is actually in line with another collaborating group in our big consortium that determined the decarboxylation time constant by time resolved infrared spectroscopy to be 300 picoseconds. So after 900 picoseconds, they told us, yes, indeed, the substrate is decarboxylated. Okay, it's now almost an hour that I've been talking. I could either go on for another 5 minutes or I could very soon stop.
No, please continue.
Yeah, so then I'll just show 2 slides that might be interesting for those of you doing crystallography and want to extract information on low occupancy states in a protein crystal. And because this is one of the issues that we do have in time-resolved crystallography, how to determine the structure of intermediate states that are actually occupied very often at a very low occupancy only. I told you earlier we determined the conformation of this chromophore in this fluorescent protein in a twisted state between 2 isomerization states. This was occupied, this conformation was occupied only to 3%. Only 3% of the molecules in the crystal had their chromophore in this conformation. So you see, it's a very low occupancy. And that's a big problem we have in time-resolved crystallography. So we have here a protein crystal in the dark state. We excite it, and only a few molecules that transit to the intermediate state. So that's what we get. The average structure is still the starting state, but what we actually want is to determine the structure of the intermediate state, the yellow state. So how can we extract the structural information? Well, this is done by structure factor extrapolation, and I don't want to go into detail here. Just mention if there's anybody among you who wants to determine, who extract information of low occupancy states from protein crystals after excitation. You can use this extrapolate program that Jacques-Philippe Gaultier and his postdoc Elke de Zeter developed, and that will be published in a few days. And in the case of our photoenzyme, for instance, you can now with these extrapolated structure factors calculate so-called extrapolated electron density maps that correspond to electron density maps as if 100% of all molecules in the crystal occupied this intermediate state. And then you can model you can construct and refine a model of the intermediate state. So for instance, here we have this substrate, this fatty acid in the dark state structure. And after 300 nanoseconds, only a fraction of the molecules got decarboxylated. And with this extrapolated electron density, we have a means to model here this hydrocarbon product and to get a model of the excited state and to see that after 300 nanoseconds, for instance, this tyrosine residue moved. Okay, so I showed you here. I showed you here an example what time-resolved crystallography can do in a structural biology project. But it is important to highlight here that in this consortium we had many different fields of expertise. We had time-resolved infrared spectroscopy, as I said, Fourier transform infrared spectroscopy, temperature-dependent synchrotron crystallography, in crystallo photospectrometry, of course, biochemistry, quantum chemical calculation, transient optical spectroscopy, in crystallo transient optical spectroscopy. All these techniques here, I didn't show you at all. So I just want to highlight here that time-resolved crystallography is a very exciting tool, but more and more it will be just integrated into the toolbox of the many other biophysical techniques that you require to understand protein structure and function on a molecular level in an integrated way. So together, we have been able here to work out in quite some detail how this photoenzyme works, which different steps it goes through. So light excitation of the FAD chromophore leads to excitation of the FAD and the electron transfer from the substrate, fatty acid, to the FAD. Concomitantly, we decarboxylate the substrate and form a hydrocarbon. Then we get back electron transfer from the radical FAD to the alkyl radical. And we identified an important residue in the active site that is involved in this proton-coupled electron transfer. CO2 that is generated by decarboxylation is transformed into bicarbonate and then leaves the enzyme. I don't want to go into the detail here. The alcan is then released and the new substrate can bind. That's our current understanding of this photoenzyme. Okay, I will come to an end here. I hope I could show you in the last hour or so how we can use large-scale X-ray facilities, in particular synchrotrons and X-ray fractal lasers, to study proteins in motion by time-resolved serial crystallography, conveniently done in a pump probe scheme. I highlighted that in the coming years, probably if you want to study slower motions, microsecond, second time scale by crystallography, you can go to a synchrotron. But if you want to study fast protein motions, you need to go to an X-FAL. I briefly alluded to how we have been able to use time-resolved crystallography to determine isomerization intermediate state of a chromophore in an electronically excited state in a fluorescent protein. I showed them in a bit more detail how we can use time-resolved crystallography at an X-FAL to determine photocatalytic intermediate in this new photoenzyme. I also highlighted how important it is to combine crystallography with simulations and also with spectroscopy. And I showed that actually by the further development of synchrotron sources, actually by implementation of serial crystallography, small crystals, in vivo crystallography, and room temperature crystallography in small crystals can now be done at synchrotrons, and that over the coming years, the niche of X-FALs in structural biology will remain the determination of damage-free structures and ultra-fast time-resolved crystallography. And with this, I just want to end with my crystallographic colleagues at the IBS in Grenoble that you see listed here, and other crystallographers who joined in applying X-FAL-based crystallography to study this photoenzyme. Yeah, if some of you might be interested in recent reviews on time-resolved crystallography at both synchrotrons and X-FALs, and I can point you to these papers here. In particular, the most recent one by Richard Neutze and Brandin. It's a very nice review. Okay, with this I stop, and I thank you very much for having stayed on the Zoom for such a long time, and I'm ready for questions if you would like to discuss.
Thank you very much, Martin. It was really a very interesting presentation with a lot of beauties about how to use synchrotron and Excel sources to develop how an enzyme works. And let me just make a comment. It's just to show all the people that having the fold of a protein is just the beginning. I mean, the details, the tiny details that actually occurs in the active sites of the proteins are much more complex to explain and to visualize even.
Yeah.
We have at least 3 questions already. Mm-hmm. And please, the people that are connected even through Facebook Live or YouTube, please write your questions. Nina, you have 3 questions. If you want, you can go on, and I ensure that Carlos also have questions.
Okay, I was wondering, when you have this capturing of the intermediate states, if you collect data at very low temperatures, like 100 Kelvin, how do you know that you're actually capturing the right conformations? Because the problem is that the free energy surface depends on temperature. So you would be getting the frequency for jumps wrong, and also the relative depth of the minima.
Mm-hmm. You're absolutely right. We don't know, and that is one of the strongest arguments to do room temperature crystallography if you can, because as you write and say, the energy landscape is often remodeled at low temperature. So we cannot be sure that the particular intermediates we determine or trap are preferred at room temperature.
Right. I have another question.
There's some music.
Yeah.
Yeah. Thanks, Enrique.
No longer.
So there's quite a famous paper now by James Fraser at UCSF. He compared crystal structures determined at 100 Kelvin and room temperature, and in particular, he looked at side chain alternate conformations. And
I remember.
You remember this paper. So if you go to 100 Kelvin, many of the alternative side chain conformations present at room temperature, they are lost. And in many cases, it's these alternate conformations, the sampling between which is so important for function. So yeah, it's a very critical point that you raise. And of course, we can, in many cases, not be sure that what we trap is functionally meaningful, yes.
And along the same lines, and then I'll stay quiet. When you showed in GFP, and also in your summary slide, where you have this tyrosine side chain of the fluorophore, you have it going from one rotamer to the other, and you catch it midways.
Yes.
So is that a transition state, or is that an intermediate state?
Well, given that this is not an enzyme, I would say it's rather an intermediate state, an excited state intermediate, a short-lived one, and not a transition state. Or how would you define a transition state for a non-enzymatic protein?
A transition state is just the local maximum on the free energy surface. And in order for it to be an intermediate, you have to have a dimple on it. So it's a local minimum.
Yeah.
The issue is whether it lives long enough so that you can actually capture it. If you can get transition states, you can define transition states for any process, because you always have a barrier to jump, no? Okay. So at the top of the barrier, that's the transition state.
Okay. Yeah, I was always, thank you for the explanations, Nina. I was always associating transition states only for enzymes, but of course you're right, and the answer is we don't know exactly, because we don't know the energy landscape of this isomerizing chromophore. Since this was still at a very short time scale, a picosecond, it could still be that we are in a coherent regime that would then mean that we have been at a transition state, because at this low temperature, still all the chromophores move in phase, they're not dephased, so it could well be that it's a transition state. But I rather suspect it's rather an intermediate state, because QMMM simulations indicated that it would stabilize after photoexcitation in this state.
All right. Okay. So then you do have a dimple.
Yeah. Most probably that's how we visualize the excited state energy landscape, with a dimple in which the excited state could accumulate for time long enough to be visualized by crystallography. But we don't know really for sure. I don't know what is, if you have any additional insight or ideas, of course, I would be very interested to hear.
I would love to see the structure to see how it's stabilized.
Okay, I can send you the PDB file if you like.
Oh, cool.
And if you have any comments or corrections or protests, it would be very nice to hear.
And if I may, just the last question.
Yes.
When you have this arginine 450 in the last example that you showed us behaving as a proton donor, that is really mind-boggling, because arginines have pKa's in the order of 12. So how do you convince an arginine to give up a proton?
Yeah, indeed, that was extremely puzzling and very unexpected that this arginine seems to be involved in a proton-coupled electron transfer. I don't know. I should know, and I should go back to the data published by our computational colleague Tatiana Domacheva, if she made any suggestions why the pK could be lowered to such an extent that it can be actually deprotonated. I don't know.
Thank you. It was a really nice talk.
Yeah, thank you, Nina, for the many questions and for the discussion. Thank you.
Carlos.
Yes, very nice talk, actually. I have a couple of questions, more in the practical sense.
Okay.
I sometimes like to do some time resolved NMR, so I know that the setup is actually really, really important. Mm-hmm. So when you were talking, I was wondering first, sometimes, I think you already answered a little part of my question, but sometimes you don't, not all your molecules are not synchronized because the light is not enough, or I'm many reasons. So at the end, I'm guessing you get double sets of peaks or something like that. So how do you recognize one from the other, or how many different species can you differentiate and still get useful data?
Yeah, no, absolutely. This is a very important point, and I'll just share again my presentation here. This example here. So here in this case, say we that most of the proteins stay in the dark state and only a few transit to one intermediate state. So indeed, by this structure factor extrapolation approach, can we tell those two structures apart? But these are only two states. And if you have more than one state, the situation gets very complicated. So we then, in these extrapolated models, structures here, we usually model only one state. But sometimes it's clear that there must be at least two states, and we try to model two states. But then it becomes very complicated, and it becomes very complicated to convince ourselves and our colleagues that we are actually know what we model here. So that's a very tricky point, as you say. It could be, if there are more than one model, one could also think of doing experiments at different temperatures to shift the equilibrium between one intermediate state and another to either one or the other intermediate state. But since these experiments are so complicated, I'm not aware of colleagues already do temperature controlled time resolved experiments to disentangle complicated mixtures of intermediate states. But you're absolutely right, Carlos, that can get very, very messy and uninterpretable.
Yeah. When I saw these slides, it seems that, yeah, you have at least some protocol to differentiate. Very nice. My second question is in the practical. So imagine that I have a protein that I want to study, and I'm trying to understand the dynamics. Is there any parameters that can tell me that if I try to do this kind of experiment, it's going to work? I don't know. In the way that I thinking this, I'm not a crystallographer. I'm thinking that if I have a mobile part that is in contact with another protein, that's not going to work. So is there, if I can look to my protein and look to the factor B or something to see if I have a chance that these kind of experiments are going to work, or is just I try it and sometimes it works and sometimes doesn't?
So is your question or your comment related to so does it focus on which type of structural changes can you actually observe with crystallography?
Yeah, I mean, I have a
not allowed?
really nice example of a enzyme. Do I just try it? I mean, or is there a specific, I can look to my crystal structure and try to see if there is a parameter that says that is more likely that it's going to work?
Well, yeah, I think it really depends on the case. And if you knowing your protein, you probably already know which type of on which type of spatial scale structural changes are expected. If you're interested in structural changes in the heart of an enzymatic active site, you can probably assume that the extent of the structural changes remain so limited that they are not disturbed or hindered by the crystal packing, even though this might not be true, of course. You could, and if you want to study structural changes in an enzyme by soaking a substrate, and you look at the packing of this enzyme in the crystal, and you see that the active site that might be accessed by a tunnel, as in the case of this photoenzyme, is blocked by a neighboring molecule, then you already can guess that, well, a substrate will not be able to reach the active site because it's blocked by crystal packing. Yeah. Then some effects of crystal packing are simply not foreseeable. There's a very nice example in the literature of a tetrameric protein, and people have done kinetic crystallography, and each of the 4 monomers had a slightly different packing environment in the crystal, and each of the 4 monomers got stuck at a different part of this catalytic cycle because their conformational freedom was differently affected by the crystal packing in a very subtle way that was not foreseeable. So they only saw it by doing the experiment, seeing that the 4 monomers react differently, and then trying to make sense of the intermediate state they trapped. And this is called crystal trapping, trapping by packing. Now, if I may add, we work on a protein at the moment, and that's probably a very extreme example. That is a tetramer, and the tetramer binds to a DNA operator, and when this tetramer is being illuminated, the tetramer falls apart into monomers, unbinds from the DNA, and allows for DNA transcription to happen. So you imagine you have, and it's a light-sensitive protein, you have structural changes on a very fast time scale at the chromophore level, and it goes all the way to the second time scale where the whole tetramer fall apart. So somewhere in between, for sure, your crystal packing will not allow this change to happen. And our first experiments indicate that you kind of hit a crystal packing wall when you illuminate its crystal. A few structural changes happen in the level of side chains in the active site of the enzyme. But then, if then helices have to move so that the conformation of the tetramer changes and eventually dissociates, this is completely blocked in the crystal. And this was rather easy to foresee that this will not happen in the crystal. Sorry for the long comment, Carlos. Thanks for the question.
Very nice, thank you.
And I have another question and a comment, and I think that we are going to end after that. But first, talking about Excel experimental setup, usually when you have in a drop several microcrystals, I mean, I'm afraid that in most of the cases you have heterogeneity in the crystals. I mean, if you hit one particularly, the order is going to give you a very nice diffraction pattern. But if you hit the neighboring one, probably you are going to have a very badly diffracting pattern coming from that crystals, or probably it's not even a single crystal, but a bunch of crystals together, which are going to give you a combination of matrices. When you are doing an XFEL experiment, how you are dealing with that, or is that part of the problems when you are trying to get a structure or a series of structures?
That's yeah, you identified this very nicely, Enrique. That's of course a big problem, and the first step in such a XFEL experiment is to produce a homogeneous sample that is devoid of the kind of problems you mentioned. Of course, if you have a big crystal to crystal variability, then an experiment in which you need to collect tens of thousands of images from the same crystal wouldn't work. Yes, so that's why in such a XFEL project you can often be busy for a year or longer in just producing the sample. Not only because you need a lot of material, but also you need to have a very homogeneous sample.
Wow.
Absolutely. You're muted, Enrique.
Sorry, I have my daughter just in front of me and she asked for something. I know that you have two. Then you understand.
I know it's lunchtime now. Yeah, I'm keeping your daughters and your colleagues from having lunch.
No. Thank you. Just a final comment, because one of the reasons that we organize and try to invite people like you to give us the frontiers, the limit, the things that are changing our minds about what structural biology is going to be.
Mm-hmm.
I mean, it's because we're kind of isolated in Mexico. I mean, we are not many groups. When the people start to start reading that you can use programs to tell you which is the structure of a protein, I mean, most colleagues think that that's it. I mean, we don't have anything more to ask experimentally to a protein. Mm-hmm. And we are not agreed. Part of the reason is because we're working with proteins, but because you are working in the IBS and you have access to all these fantastic machines, which is your point of view? Which is the future of structural biology? It's going to be integrative. You are going to still need to do experiments. Which is your opinion about this subject?
Mm-hmm. Yeah, it's a very good question that we all ask ourselves, Enrique. Of course, who will be getting jobs tomorrow, and which of the techniques will still be useful and which won't be? And it's such an exciting time that I think it's quite difficult to answer this question. Already now we see, if we look through the literature, molecular replacement has begun so much more easier now that we have the AlphaFold database that gives us, that enlarges us with the number of search models. So that's already a very clear sign that, for instance, experimental phasing, SAD and MAD, will not be used anymore very extensively, because molecular replacement with predicted structure is so easy. Now, you mentioned structure prediction, but we can of course also mention electron microscopy that is eating a lot away from X-ray crystallography. So I think it's very difficult to to predict what will happen. But for sure, if we talk about studying structural dynamics, it will be I think still a long way to really predicting not only the average structure, but also structural changes that happen. So I think the dynamic structural biology will remain largely experimental, of course, combined with MD and QMMM simulations. But the pure static crystallography will probably more and more be predictable and solvable by single particle cryo-EM. But then, I mean, as Nina pointed out, I mean, room temperature information, structural information is very important. Cryo-EM will remain cryo-EM. So I think we have a very nice set, as you put it, Enrique, very nicely, integrated approach that will grow more and more together, and that we will have to look, if we want to solve a molecular biology problem, in molecular biophysics, we really have to approach it in a holistic, integrated way, such as we tried to do with this fatty acid for the decarboxylate project. I could just maybe add something that was interesting to me. There was a paper, I think, in Nature Methods last week that showed that explained what structural biology can do in COVID research, and they listed that in the past 2 years, about 2,000 PDB structures have been entered in the PDB. Out of these 2,000, more than 1,100 have been solved by X-ray crystallography, and about 700 by cryo-EM. So we see that if you take a structural biology problem, and in this case corona, most of the structural information still comes from crystallography, and I expect that it will remain a very important workhorse to determine high-resolution structures of biological macromolecules.
Well, thank you very much. I mean, it was a fantastic talk, and also the talk that we have after, it's very illustrative. Thank you very much for your time. Thank you for being here at 8:24 p.m. France time. Thank you for all the information that you gave us. We'll see you soon. We hope to have you physically in Mexico the next year, and it will be fantastic to show you the beauties of Mexico. Also the challenge that we have, but a lot of fun as well.
Thank you very much to the three of you for hosting me. And I should maybe also add that we are always extremely excited to collaborate with colleagues from other countries and continents. And so if there's anybody of you who think you could benefit from a collaboration with us or other colleagues who are closer to large-scale facilities than you are, we are all very excited to engage in collaborations. And Enrique, you know, you have a long history of collaborations with countries in Europe, so we would be very excited.
Thank you very much, and thank you to all the audience to be here. It is in a strange moment where we are living that it seems that we are just 4 of us, but in fact there are tens of people watching this, and the video is going to remain in the channel of my institute. Then I hope more people were turn interested. I mean, see how interesting it is to do detailed experiments in proteins as you show us. Thank you very much, Martin. Thank you, Nina and Carlos.